cell biology.
So thanks a lot for having me here. I really appreciate the opportunity to come and speak to you today, and thanks a lot to Wolfgang and Ewan for inviting me. I sort of had an in as far as the program is concerned, I suppose. So I have a interesting background for the EBI, I suppose. I sort of started out in computation for many years and then moved to biology, where I became an experimentalist as well. So I sort of have a joint approach to looking at different problems, and what I've found to be the most effective for the way I like to work is to examine things from a biological perspective and then to bring in new technologies or actually develop new technologies to sort of address the questions that we'd like to answer. So functional genomics is something that I'm sure you've heard a lot about during the meeting, but essentially the way I look at it is that there's a lot of hard-won genomic and proteomic information out there, and that's been collected over many years and over really through the effort of a collective of many different research labs across the world to, sequence our genome and to provide us with information that's readily accessible that we can use actually in addressing interesting biological questions. And so functional genomics, I think, strives to make use of that information in the best way possible, either computationally or experimentally, in designing an interesting investigative study that actually reveals something about the way our cells and bodies work. So we can design experiments to look at cellular changes, for example, looking at transcription over time, looking at different molecular features like transcription factor binding site occupancy and so forth, that reveal something about the inner workings of our cell and hence the fundamental nature of life. And fundamentally, I think these experiments and these different projects are really hinged upon well-defined experiments, well-controlled conditions, which is actually a problem for people like me who like developmental biology. This is very difficult to overcome because what we're looking at in most cases are collections of heterogeneous cells and tissues that are not readily accessible in their singular form. We're looking at developmentally interesting niches and organisms that are undergoing change. And so if we disrupt that system, it's not really advantageous to looking at a biological question, even if it's technically more feasible in terms of the approaches that we take. So what we try to do is to either develop methods to overcome that or simply use inferior technologies. But in terms of functional genomics, we do have a large number of technologies that are at our disposal. These include DNA microarrays. There are many applications to this now. Obviously, it started out as singularly, RNA expression analysis, and rightly so. But now we're looking at chromatin-based profiling with different methods to look at promoter and enhancer occupancy in various cellular conditions. Recently, we're looking at second-generation sequencing technologies, which I'm sure you've heard about also from Paul Fleechik and so forth. And these are currently augmenting microarray approaches. I think in terms of this particular application area, it's really overcoming some of the approaches that we developed earlier. And there are various other technologies such as RNAi screens to assess for phenotypes and that sort of thing. And we can use these technologies to set up all kinds of interesting experiments. So looking at different cell types and states is something that I'm very interested in. Functional RNAs that don't code for proteins are now sort of on the landscape of people's attention. It's become an interesting topic. Looking at how these transcriptional changes actually affect themselves over time in terms of the cellular machinery involved, and looking at transcriptional regulation to see how that actually comes into play and sort of having this kind of machine working that actually changes your cells. So one of the technologies that we've used in the past is called chip-chip, and this is something that I've worked in quite a lot. And the idea is that we have cells that have active proteins called transcription factors. These activate genes, repress them, and so forth, and really enable our cells to carry out their genetic potential. And so what we want to do is kind of freeze the cell in a certain state and capture the DNA that's actually associated with a particular transcription factor. We do that and then isolate the DNA that's associated with the factor, label that so that it can be hybridized to an array, and then hybridize that with a non-specific sample to reveal interesting patterns of transcription factor binding site occupancy based on the enrichment of those sequences. And essentially what you get are maps like this. This was a recently completed study with Asifa Akhtar at EMBL. And this is sort of a chromosome-wide binding site profile of different transcription factors. So here are the peaks that we determined through chip-chip analysis. And this has been used to great effect by many different labs in developing these sort of large-scale maps of transcription factor binding sites related with differentially expressed genes. And so this reveals something about the global pattern of gene regulation in our cells. So now we're looking at new technologies to do this even easier. And so sequencing these immunoselected chromatin fragments is actually turning out to be a much easier way to go. And currently we're using a Selexa technology to do this. And we're generating many millions of runs, millions of reads per run, which in effect do the same thing. We're essentially just sequencing the DNA directly instead of measuring its ratio in comparison with a control sample. So then we map these onto the genome to reveal the binding site occupancy in a very similar fashion. So several people from my group, along with Simon Anders from Wolfgang's group, are now using this method to examine different transcription factor binding sites in several cancer cell-related conditions, and we're looking at stem cell technology as well. And essentially what you get back is if we map these back to the genome, you get pretty good alignments, and these sequencing reads just stack up. So the red is antisense, and the blue are sense strand, short sequence reads that are mapping back to the genome. And so this would define actually just a binding site or a series of binding sites that are associated with a particular transcription factor in the experimental study under investigation. So this is one of the binding sites that we recently resolved. And you can see that actually there are several peaks here, so we can associate multiple binding sites within one loci. And actually they map very well to the CPG island, so it does appear to be actually actively regulating transcription. When we map these back to the genome, it's pretty clear that these binding sites are actually quite specific to one particular region, and there's very little noise or non-specific binding outside of these particular control regions. Some of the other regions that we've looked at actually map to DNase 1 hypersensitivity sites, which also indicates that they're used in active transcriptional regulation. This is from Ensemble, their functional genomics build. And one of the things that we try to do quite a bit is to integrate different experiments to synthesize more of a complete picture of what we're looking at. So one of the things that has become evident in chip-chip studies is that when we resolve these binding sites, only a subset of them are probably going to represent real functional enhancers or promoters that are used at any given time. So although there might be legitimate binding sites, we want to see which ones are actually used in that particular condition. So these are going to be a mixture of promoters, silencers, and actually just random binding events as well through common consensus sequence association. So the factor might bind simply to the presence of these recognition sequences and not because it's actually being used for transcription. So what we try to do is use expression analysis under the identical conditions to reveal the affected genes in the same state. So this enables us to correlate some of these transcription factor binding sites with differentially expressed genes to reveal potential interesting regulatory interactions. So here we see different binding sites, and actually downregulated genes are indicated in blue, upregulated genes are indicated in red, in yellow. And so we can determine by the proximity which binding sites are likely to affect those particular genes. So it's not completely determined by this particular study, but if you wanted to follow that up, you would then do that. And obviously some of these long-range interactions can't be resolved using this sort of linear assay, but it gets a lot closer to what we want to do. So what we try to do is use the latest technologies available, and the Affymetrix gene chips are generally the de facto standard for this. And the setup here is the probe sets represent one or more transcripts from a given gene locus. And these are only annotated genes. And they do provide a good measure of RNA abundance, but they don't tell you anything about how the gene is actually composed. So what we found is that in different conditions, exon usage in multiple splice isoforms can actually change. So now we're using these new exon arrays from Affymetrix, which contain a different probe set for every annotated exon in the genome, either the human or mouse genome. And so then we can provide actually alternative splicing information as well as this gene-based transcript information, because we can analyze the data on multiple levels. We can collect computationally the fluorescence data over a given gene locus and determine its transcription based on the gene level, or we can actually sort of go in and look at exon-specific fluorescence signals as well. And the analysis is fairly straightforward now that new packages are out to aid us in this. So we do everything here in Bioconductor, which is managed by Wolfgang, along with Robert Gentleman, and we use standard background correction normalization procedures that are widely used in the community. But the interesting thing about this is that the microRNA, the microarray files are quite large, and actually there's a lot of annotation associated with them that you would normally not really find on a gene chip. So instead we use actually a local installation of the Ensemble core database through the R-MySQL interface to link with this directly. So we can sort of dynamically update the annotation of all the exon information on every array as it changes over time. So as the gene builds change, we can actually re-annotate the arrays in a more dynamic way. So essentially what we get are the probe sets that are represented on the array are several different types. There are sort of known probe sets that map to well-annotated, sort of RefSeq-oriented exons. And then there are probe sets that are actually in intergenic and intronic space. And these are collected from either EST reads or from previous studies on tiling arrays that have identified different transcripts that might be of interest to examine. So I don't know if you can see this in the back, but essentially what we can do is resolve exon level fluorescence measurements to try to pick apart which isoforms might be expressed at different times. And so we do see sort of a superposition sometimes of exon reads that might indicate that one or more isoforms are expressed, where we can certainly tell between one sample and another when a population of genes has undergone an exon usage change, because the fluorescence data simply changes over the length of the transcript. So I'll describe another project now that actually focuses on proteomics. And essentially this is a complementary technology that allows us to see not only the transcripts in the cell, but the protein levels and protein abundance and localization. So essentially what this does is take a cell lysis and performs an analytic centrifugation so that we can separate all the organelles actually by their densities. So then they're labeled with different isotope tags so that all the proteins are actually differentially tagged. And then these all go through a mass spectrometer. So what we get at the end are the amino acid reads that indicate the peptide being analyzed, but we also get this tag, right? So this provides a quantitation of protein levels in the cell, and also this tag which indicates where it came from based on its centrifugation and based on its organelle, so we can see where the protein was in the cell at that particular time. And actually separating these data has been fairly straightforward, but a bit noisy, because we've only got 4 tags to work with. So the dimensionality of the data is quite low at the moment, but we're working on methods to improve this. So we can easily cluster the data, and they cluster fairly well based on the isotope tags, but there is a population of proteins that actually appear in more than one organelle or subcellular fraction, and that's presumably because they either occupy both fractions or they were co-precipitated with each of the organelle fractions. But once we do that, we can essentially make these sort of data maps to cluster the different proteins based on what we believe might be an interesting or might be a relevant or biologically relevant area for them to cluster. So we can manually annotate where we know some of the proteins actually do localize, and then the ones that are in this particular region, we can assume that they're co-localizing in the same organelle. And essentially, we can also see larger complexes of interesting proteins that co-fractionate together. And this was an interesting observation based on a very simple, understanding that the ribosomal proteins should actually be together. So we would always see these together, and actually what we did is look at the ensemble families, and in doing this very this basic analysis, but using this well-annotated data, we can see that actually quite a few complexes do come out that associate with transcription and mitochondria and several different important cellular processes, and these proteins are always associated together. So along with different organelle markers, we can now actually predict where some of the proteins will localize, and this is a bit less robust at the moment, but that's actually largely because of the number of tags that we're using. So we can't really apply very interesting machine learning techniques and so forth because the dimensionality of the data is a bit low. But still, we do see some success, and actually some of the proteins that we would see distributed throughout the cell do show more of a distributed pattern than the proteins that we would see more focused, such as the plasma membrane always comes down in the same fraction, and so forth. Some of the signal sequences actually have helped a little bit in determining or predicting where the proteins will localize. However, in some of the secretory pathways, you can see this is just all over the place, so this has no real meaningful information, whereas some of the ER information has been quite revealing in determining where exactly this sort of subcellular fraction is supposed to be, if you will, as a region on this data map. In looking at the protein domains from PFAM, this has been sort of interesting in the immunoglobulin sense because they're a bit more distributed than you would find, than I thought would actually be the case. But some of them actually have been resolved in quite a tight cluster, and this is mostly done by Heidi Dvinge in the lab, who's still working on this area. So one of the things that we want to do next is actually extend this to look at more of a dynamic system. So one of the problems with this is that we're looking at a sort of a snapshot between two different cellular conditions, and the actually reproducibility between those is a bit low because we're looking at, as I mentioned before, sort of a collection of cells that are not always the same. In particular, we're prototyping this with developing embryos, and where you have a collection of cells that are heterogeneous in their state and so forth. And so we want to take cell lines directly, label them differently using a SILAC technology, and increasing the number of labels so that we get a higher dimensionality of data. We can actually label them with more subcellular fractions, so we can actually separate the data much better, and then we should be able to do more of a time course type of experiment where we should see higher reproducibility per sample and actually see the proteins changing in their location over time.
Well, just
yeah.
out of curiosity, how many proteins in total kind of come through, as it were? What?
Yeah, it's about 1,000.
About 1,000.
Yeah.
So in total, union across all the.
Yeah. Yeah. So essentially, we get between 700 and 1,200 per run, and of the ones that actually, agree between, it's probably about 900, something like that.
And to what extent, when you say that there's guys that go across all the tags?
Yeah.
Is that the same peptide, or is that the same protein?
Peptide.
Peptide.
Yeah.
So it's peptide. So all of that clustering, the clustering you showed there was by peptide.
Yeah. Yeah. So there could be actually different protein isoforms represented there that we can't really resolve yet.
Right.
Yeah. So that's actually another thing that Heidi's working on, to get the raw data from the machine and actually look at the different, because you can tell when they cross the exon boundaries which actually isoform would be expressed. So you can tell in that way, but it requires a bit more finicky analysis.
And how many spectra are unmatchable? What's the proportion of unmatchable spectra?
It's pretty high, actually. It depends on the machine quite a bit. I didn't actually realize how sort of finicky it was, but there's quite a lot of stuff that we just throw away. Say,
are we talking
40%.
40%.
Yeah. So that was higher than I expected. But actually, we're working with Yoti in Chaudhary in Center, and she gets a better, much better hit rate. And I think it's because a combination of she's got a better mass spectrometer, and also they've developed a lot of custom software to reclassify the hits and sort of do a reanalysis of them after the fact and collect more. So they do a much better job. So just to wrap up, this particular study that we're working on now, we can distinguish different proteins from different organelles, which provides us multiple kinds of information. We can see protein abundance and also localization in the cell. And so what we want to do next is essentially look at differentiating cells over time to see that basically ES cells in their resting state versus differentiated would be the sort of marker points, and then looking at them differentiating in various cellular conditions. And I'll describe one actually right now that we're looking at. So the reason that we like this idea of looking at ES cells is simply because it's the easiest way to get at mammalian development in a way that's sort of reproducible and accessible to developmental biologists. Essentially, it's a cell line that are explanted from the inner cell mass of a very early embryo. And so this is an isolated window into very early developmental events. They have several characteristic properties that are interesting from a cell culture point of view, as well as from a developmental point of view. So they undergo indefinite division in vitro. So we can essentially maintain them to give rise to identical daughter cells indefinitely over hundreds of different divisions. And they have this very interesting property of nuclear potency. So this is the main feature that sort of the news media latches onto. They have the ability to become any cell type in the mature organism, in theory. We don't have the protocols to do that actually directly, but by differentiating them into several different branch points that we know will give rise to all of these different cell types, we know that that's theoretically possible. And we can induce them to differentiate into those specific cell types artificially, which is actually the whole point here. So we want to induce them to differentiate and then observe them changing over time using this technology that I've just described. And so we can think of actually every cell division as being a choice between differentiation and regeneration. So essentially, they're derived from the very inner cell mass of the blastocyst, so the developing embryo. And we can see that they give rise to the three major cell types of the adult organism. And if I just sort of give you this slide instead, essentially here's the inner cell mass. ES cells come from here. The trophectoderm cells come from here. ES cells cannot give rise to an entire complete organism because when we culture them, they're isolated from this blastocyst. So that all these cells here actually will support the developing organism by giving rise to sort of the extra embryonic tissues that would normally support the development of the embryo. So ES cells, when they're isolated, have no potential to develop a person. Okay, so these are cell lines. It's a tool to examine differentiation. And essentially, there are several different pathways that we actually know are involved in differentiation events. But as you can see, they're sort of skeletonized here because we know some of them are complete or pretty complete. But that's only because when a certain factor was found actually to affect differentiation or regeneration, if it interacted with a well-characterized pathway, we can sort of fill that in, right? There are others where we know, particularly NANOG, as a transcription factor that's quite important for ES cell maintenance, but we don't know what it interacts with, and so this is just a question mark. We have no idea actually what this is doing. It's a black box. And so these are the type of molecular systems that we want to investigate. And when we even look at cell maintenance, the simple question of what actually maintains self-renewal in culture. In the mouse, it's getting there. It's getting to be well characterized, but it's not there yet. Humans, it's nothing. It's seriously have no idea why this is happening. There are analogous factors that seem to contribute, but it's not really something that's well known yet. So there are a lot of open questions. Even though we can differentiate them artificially, that doesn't mean that we know what's going on the molecular level. So the characteristics of ES cells that we like are that they're biologically interesting and that they're derived from this very early embryo. They're clonogenic. They have sort of an unlimited cellular resource that we can use to study, and they're pluripotent. So what we're essentially studying at the moment is a combination of differentiation and early lineage commitment. So what we found is that ESL populations are not stable when we actually sort of maintain them in culture. Some of them always differentiate to various cell lineages, and we're not really sure why. But most do remain in self-renewal, so we can still do the experiments. But we know that there's a constant battle between sort of poised to differentiate at any given time. And when we do this deliberately, so differentiate them with various cytokines and so forth, only a fraction of them actually do that. And that's very interesting from a biological standpoint, because they're all exposed to the exact same environment. There's no reason why some should respond differently than others, unless they were in actually a different state. So we're not really sure why that's happening. But in a practical sense, as I mentioned before, this heterogeneity in different cell states can actually cause a lot of problems when we start to analyze genomic data, because it starts to sort of introduce the superposition of many different states, and so it's not a focused analysis. So in the long term, so what factors influence actually that lineage selection? Can we determine that? And two, in a more practical sense, can we use a more reliable system than plain ES cells to actually study differentiation? So we're now using neural stem cells, which are derived directly from ES cells, but actually are more stable, and I'll show you why. So ES cells are maintained in the mouse in leukemia inhibitory factor, and can sort of regenerate indefinitely. Now, when we release that condition, so we remove leukemia inhibitory factor and let them differentiate, they spontaneously differentiate to all the three different primary cell types of the mature organism, but there's a definite preference for the neural lineage, and no one really knows why. And there are sort of feeble explanations that say, well, neural cells develop first or something, but no one really knows why that's happening. And actually, you get about 50% that will develop to neural, versus the other two are much lower than that. And so we're sort of using that to our advantage and saying, well, maybe that's a bit of a default pathway that we can actually exploit in developing a bit more of a stable system to work with. So ES cells can be differentiated to these neural epithelial precursor cells. And the reason they're turning green is that we've labeled this particular marker with a GFP, with a fluorescent tag. And so as the cells transition from this state to that state, more of them will turn green, and we can actually assess that quantitatively. So as ES cells differentiate to neural epithelial precursors, we can then differentiate them further to neurons, astrocytes, and oligodendrocytes, the major cell types of the nervous system. And so the question is that from that transition point, can we actually stabilize this interaction, this neural epithelial state, into something that's more like stem cells? This is very transient, but we'd like these to self-renew. We would like these to be a tool, actually, that's a more stable system to differentiate them to neurons than ES cells would be. And Steve Pollard from Austin Smith's lab has accomplished this in a very elegant way, and essentially in less than 2 weeks can differentiate ES cells through this neural precursor stage onto these sort of expansion stage of very homogeneous cell types, which then we're calling neural stem cells because they are clonogenic and self-renew. So essentially, we go from ES cells to a neural stem cell in a very short amount of time, and it's actually a very easy protocol that involves only a few different growth factors and conditions. And their differentiation can actually be very well characterized in terms of their morphology. They're all pretty much the same. They go through these transition states where they self-renew and just kind of stay there, dividing in culture indefinitely. So we can then take these out of self-renewal and then differentiate them on purpose in a much more reliable way than would be possible with ES cells. So this is basically their morphology. They have these sort of bipolar extensions here that would be preaxonal. And then this is about after maybe, I'd say 6 weeks of growth. So these are human, and so they take quite a long time. But actually, this is a very, very homogeneous cell culture. And you can see that they're actually growing on a monolayer, so that there's no possibility that we're missing differentiation events. So essentially, when cells are grown in suspension cultures, a lot can happen that's not observed. And when cells are actually grown in an adherent culture on top of a plate, you can see that they're all uniform and so forth, and you don't have a lot of spontaneous differentiation that can essentially take over. So one of the very interesting things about these NS cells, neural stem cells, is that even though they were derived synthetically, actually they express all the markers that you would normally see in a cell type called radioglia, which is the developmental precursor of all of the neural system, central nervous systems. So essentially, all the markers that you would normally see lighting up are there. So we can't really call them radioglia because they're actually derived in a synthetic fashion, but in fact they look like a really interesting biological cell type. They're very uniform, and actually one of the interesting things about this is because they actually express these known markers that have antibodies and have tools developed against them, we can actually look at different cell types within the population and then select them out. So using a technology called FACS, or fluorescence activated cell sorting, we can select different populations of these cells to reduce the heterogeneity even further. So essentially we have a very reliable system to differentiate them, and then we can select them out of culture in order to do genomic analyses with. So this becomes a very interesting technology to use because they remain multipotent. So essentially we can remove them from the media that keeps them in self-renewal and then induce them to differentiate to neurons, oligos, and astrocytes even after hundreds of different cell divisions. So this is a much more reliable system to use than regular ES cells. And actually, interestingly enough, so we can do this in human and mouse, and it's an identical procedure, and it's exactly the same. So again, we're not sure what's happening at the molecular level, but the protocol is identical. And we've even used this protocol to derive human cancer stem cells. So taking human glioma samples from various patients in pathology labs, we can actually apply the same differentiation protocol to this and excise or derive stem cells that were actually present in the cancer tumor itself, which is quite a unique thing. So revisiting this slide, these are the things that are important about ES cells. And essentially, NS cells are very similar, except that they're not pluripotent and multipotent. They only derive neural cells. But they have all these advantages, right? So they grow in a uniform monolayer. They're inherent. The morphology is quite predictable. And so it's a much better tool, actually, for genomic research. So some of our current projects include transcription profiling. So we're differentiating them and then using a variety of different technologies to assess their transcriptional population. So this includes the exon arrays I talked about. We're now doing some sort of preliminary selective sequencing of transcriptome populations to see how we can compare that to traditional microarray technologies. We're looking at microRNA expression as well. So essentially using microarrays and real-time assays to assess microRNA level and then relate that back to transcriptional levels computationally to see if we can actually see message levels depleting over time. And then one of the more interesting projects involves nuclear reprogramming. So essentially one of the things that's been publicized recently is the conversion of mouse and human fibroblasts or skin cell-derived cells to a state that resembles an embryonic stem cell state. And we're now doing this with the NS cells, and it looks really good. So essentially, there are several tests for pluripotency, and they all pass. So they are indistinguishable in a sense from ES cells. So we can take embryonic stem cells, we can differentiate them to neural stem cells, and then we can go back, which is normally not something that is found in biology, in vivo. But by using a specific combination of transcription factors, we can actually cause that process to reverse back to a state where these cells can be induced to a pluripotent condition again. And in doing that, it's kind of the reverse from what I described earlier. And we're doing ChIP-seq experiments with key transcription factors like OCT4, NANOG, some of the ones that I talked about earlier. And this is all with Solexa technology now, and we're interacting with people like Paul Fleechik and Ewan and Wolfgang to analyze all these data at EBI. And we're looking to see how actually that process is reversed in terms of the gene regulation that underlies the morphological change. So we know these transcription factors are quite important for that to occur in the forward direction, if you will, right? But how do they actually collaborate to reverse the process and erase all of the events that have taken place up until then to revert cells back to an ES cell state? So that's quite a different problem than what we've looked at before. But it's a really exciting project, and that's being head up by Molly Salmon-Devan in my group, along with Austin Smith's lab. So I'll just wrap up with acknowledgement to collaborators. Basically, we work a lot with Austin Smith, Steve Pollard, and Jose Silva at the Center for Stem Cell Research here. Alfonso Martinez-Arias and Penny Hayward are instrumental in developing a lot of the mouse prototypes that we use. The LOPA technology was developed in the laboratory of Catherine Lilly with Denise Tan, and we've done all the analysis on that with Heidi Devinge in my lab. And Kaidi Tamoya does a lot of Solexa analysis for her projects, along with Simon Anders from Wolfgang's group. And Molly's heading up the nuclear reprogramming project, which is just starting now. So thanks a lot for having me, and I appreciate it, and I'll take any questions that you might have.
You talked about how the ES cells can differentiate into different kinds of cells, and
Yeah.
it's not understood what goes into that decision.
Right.
So I was wondering if people have looked at how correlated the distance, so for a given ES cell, how close it is to other ES cells, and whether the proximity
It matters.
somehow.
Yeah, it does matter. And actually, when we differentiate other kinds of cells in a suspension culture, which is why we try to avoid that, there's this sort of wave of differentiation through the culture that's somehow mediated by signaling molecules. So they collaborate, in other words, and influence each other through some extracellular signaling that's not well understood. And it's probably related to the growth factors that are now being isolated to do this artificially. So that's one of the major problems in stem cell biology, is that you know where you want to go, but it's really hit or miss in terms of trying to differentiate cells in vitro, because we don't know the molecules that are involved. And in fact, the major one, leukemia inhibitory factor, that's involved in suppressing differentiation, was only recently discovered. And that was the major constituent that was being expressed by these feeder cells that were being used to support their regeneration artificially.
So can you get enough ES cells to do mass spec with the exuded factors?
Yeah, because we can really clonogenically continually regenerate them. Now, we have to be very careful that you do have a homogeneous population, which is really difficult in ES cells. But that's kind of one of the reasons why we're moving to this other one. But yeah, the level of material is very important for mass spec. Anyone else? No? Okay. Well, thanks again.